So we took a large panel of mutant mouse embryonic stem cells, and the panel is made up of different stem cell lines, each line with a different gene mutated in it. And we took those stem cell lines and we screened them, look at basically the gene function for differences in sensitivity to rabies virus. And the way we did that was we were able to utilize a rabies virus that was labeled green with a green fluorescent protein, and so when you look at it under the scope, it literally glows green. And so we were able to infect the stem cells, and we were able to differentiate the stem cells even, because if you think about stem cells, what's remarkable about them is that they can form all the different types of tissues in the body. So we made neurons out of them because that's one of the major cell types for rabies virus. And so we grew up the neurons and infected them with rabies virus, and were able to measure how much green protein was in each cell line after infecting it. And we did that over time. We would measure at 24 hours, at 48 hours, and at 72 hours, and look at those with more green were more sensitive to the virus, and those with less green were more resistant to the virus. And so the reason this is really cool is, and the reason it's never been done before is, first of all, to get a panel of stem cell lines, we took literally thousands of cell lines and differentiated them simultaneously. And we had to develop some tissue culture techniques to get that online so that we could look at so many cell lines at the same time. And another reason no one had ever tried this before was because it was questionable the way these stem cells were made. The mutations were made as we knocked out basically one copy of a given gene, and everybody has two copies of every gene. And so by knocking out one copy, it was questionable as to how bold or dramatic a phenotype we would see, because you're still expressing half as much as you normally would. And we validated that, or we made sure that we were able to see a phenotype by taking genes that were known to be involved in rabies virus infection, such as known receptors that bind to the rabies virus. And then we screened those cells and showed that we were able to see an effect. And so after we validated it that way, then we went and screened thousands of cell lines. And basically what we found in the end, when it's all said and done, we found about 63 different novel genes that have never been implicated before in rabies virus. Several of them have been implicated in other viral infections, but never rabies before. And so these 63 genes made the cells either more or less sensitive to the rabies virus. And why that's important is because now we have new genes and new pathways to look at to analyze viral infection or bacterial, well, viral infection in this case, and it gives us new drug targets to look at and new pathways to look at. And more of an overarching implication is we were able to, while we specifically looked at rabies virus and neurons, this can be applied to virtually any type infection, whether it's bacterial or viral or toxin, or even drug interactions within a cell, or radiation sensitivity or resistance. So we can do a variety of assaults on the cell and look at what genes are involved in protecting or making the cell resistant or sensitive. Another nice thing is, because they're stem cells, not only can we can screen stem cells, which are good for toxicology. We can screen neurons, as we did in this paper, but we can also differentiate them into other cell types like cardiac tissue or gut epithelia or renal tubular cells, or whatever cell type that we want to differentiate them into.